Halo semua, Assalamualaikum.
Waalaikumsalam, Bu.
Waalaikumsalam, Bu.
Gimana semua? Apakah sudah hadir? Atau masih ada yang sholat? Reno, ada yang tidak hadirkah hari ini selain Reli?
Ada Julia, Bu, sakit katanya.
Oh, Julia sakit. Oke, jadi Reli sama Julia. Terus yang lainnya gimana? Kita bisa mulai atau masih mau menunggu teman-temannya?
Mungkin bisa menunggu sekitar 5 menit lagi, Bu.
5 menit lagi. Oke. Baik semua, sekarang sudah waktu menunjukkan jam 15.50. Apakah kita sudah bisa memulai kuliah hari ini?
Ya, Bu.
Oke. Hello, Miss Christine.
Hello. Hi.
Hi. The student is all ready to start this these courses for today.
Okay.
Okay.
No problem.
Okay. Let me open first, Miss. Oke, semua. Assalamualaikum warahmatullahi wabarakatuh. Jadi hari ini, jadi hari ini kita sangat beruntung untuk mendapatkan tambahan pengetahuan di mata kuliah kita, yaitu Biologi Sel dan Molekuler dari salah satu dosen di Universitas Trinity University of Asia di Filipina. Beliau itu merupakan salah satu dosen di Departemen Biologi dan memiliki gelar master di bidang mikrobiologi di Universitas Santo Tomas, Filipina. So, good afternoon to all. Today, we have the opportunity to get additional knowledge in cell and molecular biology courses from one of the lecturers from the Philippines is Mary Christine Eikada. Currently, Ms. Christine is one of the lecturers in the Department of Biology at Trinity University of Asia in Philippines and holds a master degree in microbiology at the University of Santo Tomas, Philippines. Jadi kita tidak usah lagi berbuang banyak waktu. So let's warmly welcome to Miss Christine for today's lecture with the theme of the implication of molecular biology in agriculture. Ma'am?
Thank you so much for that introduction. So I would like to start now our discussion for agriculture in new generation, biology, molecular implication for the agriculture. Okay, let me just check the report. So again, thank you for the opportunity for inviting me to discuss this actually relevant nowadays. So let's start first with the currently news in terms of the food. Currently the world is facing several issues, and one of those is the soaring population, where the agricultural production is unable to meet the growing demand in terms of food availability or production as well. And also, the pollution is actually becoming severer, and the quality of the agricultural products continue to decline. Therefore, the agricultural production has relied on hormones as well as chemical fertilizers, and there are abuse of pesticides, increase the dosage and residual pesticides. Okay. Now, the agricultural production has become a very important issue in the construction of national economy. And that is the reason why, with the development of molecular biology, some DNA-based technologies have shown great potential in promoting the efficiency of crop breeding program, as well as protecting germplasm resources and improving the quality and outputs of agricultural products, and also protecting our economical environment. Okay, so making the roles in terms of, or the making roles of the DNA in modern agriculture more and more important. And that is the reason why I entitled this discussion as Agriculture in the New Generation, Biology Molecular Implication for Agriculture. Okay, so let's start with the basic information regarding the genetic material DNA. So we all know that DNA is the main genetic materials for all cellular organisms, wherein preserving DNA itself is one way of preserving germplasm resources. Okay, so when we say germplasm, this is a term used to describe the seeds, plants, and other parts of the plants. Okay, wherein we use this germplasm in terms of crop breeding, research, as well as conservation efforts in terms of the plant itself. Okay, so now With the biotechnology, or with the development of biotechnology, as well as molecular biology, it makes us possible for us to regulate or even control the plant traits. And by using the DNA sequence information, such as the structure, the function, as well as the mechanism, we can actually do anything with it. Okay? Now, DNA technologies based on molecular DNA molecular markers, transgenic technology, and gene expression have widely been used in agricultural production that showed great potential in improving agricultural yields as well as quality. And this actually results in reducing the loss of various biotic and abiotic stress that cause, and promoting the utilization of germplasm resources and improving the breeding efficiency and strengthening the regulation of the plant growth, indicating that we are actually not into a traditional kind of cultivation in terms of plant products. Okay, so there were many reports about application of various DNA technologies. One of that is the vaccine that we have, in terms of mRNA technology. And in terms of DNA application in agricultural product is now very evident in some countries. And the DNA technologies have been constantly developed and updated, which make it difficult to introduce every DNA technique. So indicating that there are numerous DNA technologies wherein they have different agricultural applications. And I have here, okay, the common one, the four different agricultural applications in terms of DNA technologies. Okay, so of course I mentioned about the DNA, so indicating that since the advance of the first generation molecular marker technology, wherein it actually introduced the restricted fragment length polymorphism, okay, or also known as the RFLP, okay, and the polymerase chain reaction, or PCR, as we are familiar with nowadays. In 1980, there are various DNA markers were developed and used in biology research. Okay, so this includes the DNA random amplification polymorphic DNA, or the RAPD. The second one is the single nucleotide polymorphism, or also known as the SNP. The third one is the amplified fragment length polymorphism, or AFLP, and the last one is the sequence-related amplified polymorphism, or SRAP, or SARAP as we call it. Okay, so compared with those traditional morphological markers as well as cell markers, or also known as the cytological markers, and of course biochemistry markers, the DNA markers nowadays have much information and higher polymorphism. Okay, and we can work without the influence of plant organs as well as developmental stages and various environmental factors. Before kasi, when we do crop production, or we're going to screen the crop production, we actually check the temperature, what is the right pH of the water, if you're going to supply that to your plant breeds. So here, we only rely with the DNA structure. Okay, so actually, this kind of technologies is already been widely used in identification of crop varieties, as well as conservation and evaluation of germplasm. So also, these kind of technologies, they are actually now used in different analysis of genetic adversity and evolution, which is very significant nowadays, and construction.
Excuse me, Miss. Excuse me. I'm sorry. Can you show us can you show us your material?
Sorry, I forgot to share.
It's a problem. It's okay.
Can you see now my presentation?
Yes, very clearly. Thank you, Miss.
I'm very, very sorry for that. So let me just have a short recap from the previous one, wherein I discussed those problems that we encountering nowadays, especially with the increase of population, and there's an increased demand in terms of food products. So of course, addressing the food products, we are now relying to the improvement of agricultural products using DNA technologies. Okay, because they actually inform different researches or publication indicating that there are advantageous there are different advantages if we're going to use DNA based technologies. So these advantages includes promoting efficiency of crop breeding, as well as from protecting germplasm resources and improving the quality and outputs of agricultural products. And of course, at the same time, protecting our eco environment. So this, in terms of environmental phenomena, is actually very relevant nowadays, no? So that leads me to my discussion entitled Agriculture in the New Generation, wherein we are focusing on the use of DNA. So DNA is actually very useful nowadays, not just in medicine field, but also in agricultural setting. So there are actually new technologies already introduced, not just in the market, but also in the field of biology. And they're now introducing the use of DNA, okay, using molecular markers. Okay, so let me just erase some of my writings here. Okay, so what we have here are the after the discovery of the DNA by Watson and Crick in 1980s, it actually developed into more useful technologies. So this includes the DNA random amplified polymorphic DNA, or the RAPD. We also have the single nucleotide polymorphism, or SNP, and also the amplified fragment length polymorphism, or AFLP. And the last one, the common one, is the SRAP, as we called it, or SRAP, the sequence related amplified polymorphism. Okay, so instead of using those traditional morphological markers, like the physical characteristics of the plant, and also relying to different plant cells, as well as those biochemical or biochemistry markers of the plants, we are now into DNA level. Okay, when we say DNA level, we actually use the different genes that can promote the plant production. Sorry, let me erase that one. Markers. Okay. So now the question is, what are the different agricultural applications of this DNA, wherein it will utilize the DNA technology in plant production? So let's start first with the cultivar identification and analysis of seed purity. Okay, so for a long time, plant cultivar identification as well as seed purity tests were mainly based on phenotypic traits. Okay, however, the rapidly increasing of crop cultivars and the close evolutionary relationship among some cultivars and the confused varietal names of those plants, especially those local names, so the phenomenon of the homonym and the synonym have been making it more and more difficult to collect information and classify plants. Okay, so with the use of DNA, we can actually identify, okay, the plants itself without relying to those physical characteristics of a plant. Okay, all we have to do is to isolate the DNA, because the DNA itself from the plant actually have high specificity. They also have a selectivity process. And of course, identifying is actually very simple, in terms of using the DNA, and it will give you an accurate identity of the plant's germplasm and also the genetic stability of the plant products. Okay, so for example, the DNA molecular marker technology is actually being used nowadays in identification of cultivar as well as seed purity in terms of the rice, as well as the maize, okay, as well maize, these are corns. We also have the ability to identify specific cotton plants and of course grapes and among others. Okay, so this kind of identification already been used by different researchers such as Zhang et al. In 2012, Zhao et al. In 2011, and Leng et al. In 2012. No, so they easily identify the plants itself. Another one, okay, the preservation and evaluation of plant germplasm resources are very important for keeping the rich diversity of breeding parents, as well as genetic-based and rational using the germplasm efficiently and rationally. Okay. Now, with the use of DNA molecular marker technology, it will provide an important technical mean in terms of preservation, as well as identification, evaluation, mining, as well as innovation. Okay, so there are two studies by Ram et al. In 2007 and Moncada et al. In 2006, wherein they analyzed the genetic diversity of field crops such as rice and grapevines, wherein they use the marker known as the SSR marker. Okay, SSR marker. So their study actually provided a very important theoretical basis for the utilization of the rice as well as the grapes. Okay, so we're in by using the DNA molecular marker, no, not only can we protect the genetic integrity of germplasm resources, but we can also maintain the minimum breeding population and seed amount, okay, that we can use for the plant production. And also, we can screen, okay, the important germplasm, and we can preserve large amount of germplasm. We can actually further use, no, those germplasm if we need, especially with the increasing population. Okay. Another use of the DNA is the heterosis prediction. Heterosis prediction can greatly reduce the blindness as well as increase the pertinence of breeding. Okay, it is important to improve the breeding efficiency of a plant and shorten the breeding process. Unlike in traditional processes, it will take a long time for you to breed a certain food crops. Okay, so here it will actually take about less than a year if you successfully use the DNA marker. Okay, so here by the DNA marker technology, we can detect the positive markers, or also known as the positive locus. I cannot write anything. Sorry, this one, the positive locus related to heterosis in any period or organ of the plants. And then we can predict the heterosis based on the heterozygosity of the locus. Okay, the specific character for you to improve the food crop. Okay, so this technique, the heterosis prediction, okay, the DNA marker overcome the shortcoming of the isozyme-based heterosis prediction, which is actually has too much restriction to be widely used in different applications. So in the recent years, the use of these molecular markers has made some progress in the prediction of heterosis, especially in corns, in rice, as well as in cotton, grapes, and other crops. So for example, the study of Percy et al. In 2006, they found the heterosis of ball number as well as the weight of a single cotton, wherein it showed a significant relation in terms of the genetic distance of the molecular marker. Okay, so another study of Solomon et al. In 2007, they reported that the genetic distance of the molecular marker has a significant positive correlation with the heterosis of grain weight. Okay, so the relationship of these plants can actually improve the breeding of your plant crops. And of course, the most significant nowadays, the construction of genetic map. Okay, I believe you are familiar with this one, wherein it actually tells us about the genetic map as the chromosome linkages, okay, that uses the chromosome recombination and exchange rates as the relative length and genetic markers as the main body. Okay, so with the use of this genetic mapping, it has a great theoretical and practical significance, such as quantitative trait loci or locus. Okay. We also have the marker-assisted selection, the gene map-based cloning, which is actually relevant previously in the earlier years, and also the comparative genomics, which is actually what we have nowadays, no? The omics era. Okay. So the genetic map constructed by the DNA molecular marker techniques actually give us a chance to analyze the linkage relation of the genetic locus at the molecular level directly. Okay, so the genetic loci or locus we can get from the genetic map that is far from what we can get from a traditional kind of markers. Okay, yung mga physical characteristics of the plants. Okay, so at the same time you can actually with the use of the markers like this one, it helps you to do gene mapping, cloning, and marker assisted breeding. So with this one, what I have here is that molecular marker can link to the target phenotypic traits loci, by which we can locate the important agronomic traits of related genes. Okay, so when we identify, okay, the most important traits to produce great products, great food products, it will provide the convenience for the marker-assisted breeding and map-based cloning of these genes. When we clone the genes, we can actually increase the production of the plants. Okay, so on the basis of the precise location of the target gene by using the DNA molecular markers, so the breeders or the researchers can actually take advantage of this kind of technology. You can produce a lot of application when you identify that gene. Okay, so that is the reason why, in terms of gene, traditionally, the growth and development condition and the phenological periods of crops are the main reference basis for agricultural production. But if you identify already the gene specific for these characteristics, you can improve actually improve different kinds of species. Okay, and with the use of gene expression, this information can be collected, okay, through observation and also recording the phenotypic traits of the crops rather. Okay, so indicating that this method has played significant roles in traditional agricultural production. Okay, that is intuitive, simple, as well as effective. Okay, and another application of the gene expression is that the appearance of those phenotypic traits. When we say phenotypic, this is what we can observe using our naked eyes. Okay, so the appearance of those phenotypic traits, they always lags and cannot give feedback the real growth and development situation of the crops as well, of the crops in terms of their environmental factors. Okay, so what's worse is that it's always too late for us to take remedial measure when the negative phenotype appears in terms of observation. So here in gene expression, you can already identify the marker that will exhibit bad characteristics. And you can actually eliminate that from your genes, from your DNA structure. Okay? So with that in mind, okay, the development of modern molecular biology technique will provide an extremely rich and even redundant gene information to the researchers. Okay, so even though gene expression profile has been widely used to address the relationship between ecological influence or the disease phenotypes, no, so the information is still limited to laboratory experience. That is actually what we are experiencing nowadays, no. So with this in mind, it is necessary to apply the crop growth and development related to gene information to agricultural products. So the techniques as well as the methods that are available to convert this gene information is actually can be convenient and easy access information, which is actually very urgent nowadays. There's the reason why in terms of studying biology, we are now into the gene level. What I say to my students in terms of treating any diseases, diseases that can affect the plants, the animals, as well as humans, we are now in DNA level because we can easily identify those good and bad traits. Okay, we can actually, if we identify the bad traits, we can eliminate them. Okay. So as mentioned, we are now focusing on molecular biology using the DNA markers, as mentioned. There are high necessity and feasibility to apply gene information, especially in agricultural products. So people can use genetic information to observe as well as monitor the growth status of crops and also provide guidelines for the field management. Okay, so we can actually identify what can actually affect, let us say, environmental factors in terms of having weather, bad weather. Okay, it can actually give us what we can do on our crop production, and also we can actually improve the efficiency of agricultural measures. Okay, such as fertilization as well as the irrigation. And these are actually essential if we are trying to improve the food production. Okay, and also regulate the maturity and growth habits of our food products or food production. Okay, and also other important growth process of crop where it's actually needed. Okay. Okay, so in terms of the genes, in terms of the DNA marker, okay, we have what we call the plant or crop phenology. Plant phenology is the appeared time of various iconic morphological characteristics. Okay, so what are these iconic morphological physical characteristics of a plant, such as seed germination, in terms of the leaf production, okay? Ano pa ba? We have the fruit growth as well as the development status in a plant life cycle, okay? So the plant or crop phenology is the reaction of growth together with other environmental factors as well as those development and activity of plant response to different climates, okay? So especially nowadays. So by observing phenological events, we can grasp the seasonal variation of crop. Okay, guide crop planting. We can also do cultivation management and disease pest control if we only identify those factors affecting the plant growth. However, observing and recording the phenological period need to spend a lot of time, and also we need a lot of workforce or manpower and also financial resources to identify what are the good environmental factors for you to have an increased production of the plants. Okay, but here, if we're going to use the genes, the DNA, we can actually address this kind of limitation. Okay, so at the same time, uh, the climate change will not be a problem to us in relation to agricultural production. Okay, so indicating if we're going to apply the DNA technology that is already available nowadays, we can actually use it on our advantage because we have three reasons. Using this DNA technology, they're actually simple to use. They are more efficient to use because you don't need a lot of manpower to observe those plants while they're growing. Okay, you already know what are the possibilities if they grow with specific time period. And then at the same time, it will cost us less. It is actually a good, it's actually a very good advantage if we're going to apply this kind of molecular activity to our agricultural products. Okay, and not only that, no, in terms of the growth of the plants, no, the gene expression information can also be used to predict the effects of environmental stress, especially on the growth and development of the crops. Okay, so especially nowadays with the changing temperature, environmental temperature, with the extreme weather condition, this will not be our problem anymore. Okay? Because we can already manipulate the DNA structure of the plant. And this will be convenient for people to take appropriate management measure to address the problems. Okay, so unlike before, no, in the past, people will actually grasp with the growth state, no, under the stress that is brought to the plants, no. So, for example, the ozone layer, no, in 1990s, they mentioned that the ozone have a loophole that is responsible in the increase of temperature of the atmosphere. That actually leads to drought, no? And then at the same time, here in the Philippines, for example, as we are actually a tropical country, we experience extreme flooding because of extreme weather. Okay, so this kind of phenomena is actually a problem for us in terms of food products, no, in terms of food production. So here we already have actually this kind of gene application is already being used here, especially in Laguna, wherein they already they know, no, the plant already know how to prevent that kind of environmental stress. They will not be affected by those environmental stress. So another one, no, there are many genes of crops involved in transcriptional response to stress such as drought, high salinity or high salt concentration, and also low temperature. So the expression level of these genes can increase as well as decrease affected by stress condition. So therefore, through the expression information of these resistant related genes, researchers will able to know whether the crop encounter unfavorable growth conditions because it will affect the DNA itself and take timely remedial measure. And it will only take a short period of time to address that kind of problem. You don't need to go back again to the beginning so that you can actually solve those kind of problems. Okay, so another one is that in terms of the fertilizers, okay, in the past, determining fertilization time as well as fertilization types and the amount of fertilizers in agricultural productions are generally implemented based on crop growth as well as phenology. Okay, so generally experience operation and will be greatly affected in terms of geographics and environmental condition. Okay, So these kind of practices not only failed to achieve the best benefits in terms of the fertilizer application, this also resulted in the waste, okay, of those fertilizer and environmental pollution, and that affects our environment. Okay, so therefore, how to invest a reasonable input in terms of the nutrients, we can actually rely in terms of this one. So in terms of, for example, you have the BT corn. We have this kind of product here in the Philippines. BT stands for Bacillus thuringiensis. It is a bacteria wherein they incorporate the genes responsible for as a pesticide. We call it as biopesticide. Okay. In the Philippines, in earlier years, their practice is that they're going to spray chemical fertilizers to the plants so that can contaminate the corn itself. Okay, so that is actually have a negative impact to the humans. So what they did is that you have here the corn, for example, this is the DNA of the corn, for example, and then you let me repeat that one. You have here the PT corn. Okay, so it's a combination of bacteria and the corn. So what they did, they get a specific gene from the Bacillus thuringiensis that can be used as biofertilizer. Okay, so when they cut the gene responsible for that, they incorporate that to the DNA of the corn. Okay, so they actually process it, and then it produces what we call the BT corn. So here, the corn already have a natural fertilizer. You don't need to spray chemical fertilizers, and that is a good way to protect our environment. Okay, so you don't need to spend another money to have fertilizer, to buy fertilizer. The corn itself already have that fertilizer. Okay, so here, another positive or advantage of the DNA technology, it actually makes possible for people to understand the effect of fertilization, promoting plant growth and development at the molecular level. Okay, so for example, Chu et al. They publish a research relation to the effects of fertilizer. Okay, so wherein they found out that they can determine the effect of fertilization by expression of some genes related to nitrogen metabolism in grapes. So indicating the nitrogen are being processed naturally, not in other chemical base. Okay? That is actually healthier for humans, for the consumption of the grapes, no? So those are some advantages. Now, another advantages, okay, of using DNA-based technology like the transgenic technology is that we can actually improve accurately the crop quality as well as the yield, and we can actually improve the stress resistance of those plants against disease resistance. No, we also have the ability to improve the plants in terms of being insect resistant, cold resistant, and herbicide resistant. Especially here in the Philippines, we have a place called Baguio, wherein this is where we have a high production of fresh produce. Okay, so if there's an increase of temperature in Baguio, there will be a decreased number of plant production because the plants cannot grow with extreme temperature, cold temperature. So what we have here, if you're going to incorporate a certain gene that can withstand cold, we can actually produce plants even if there is extreme cold weather. Okay, so of course insects, no, there are pests. What you call this? Pests. We call insect pests for the plants. We have that in coconut plantation somewhere in Mindanao area. So what they did, instead of using chemical pesticides, okay, they also incorporate some genes from some bacteria that have a natural insect resistance. So the plant itself produce a natural way for them not to invade by those insect pests. Okay, so you don't need you will eliminate the eating of those chemical pesticides. So that is a way also for the humans and also for the environment, because we eliminate chemical pollutants, especially in lithosphere. So another advantageous of the use of the DNA markers, we can actually control the agricultural diseases and insect pests, as mentioned a while ago. And we can actually reduce the cost of agricultural production, because the seed itself of the plant, you don't need to spray every now and then the chemicals. No, the plant itself produce that substance that can actually combat those insects or other pests. Okay, we can also improve the effect of disease and insect control. Okay, as mentioned a while ago, the plants already have a marker, wherein that marker is considered as a trait that can protect them from any kinds of insect pests, other fungal plant pathogens as well. Okay, so and also we also reduce the pesticide pollution, because pesticide can actually harm not just the land but also the water or aquatic resources. Okay, so it also helps us in increasing crop production, because we don't rely more on manpower. We only rely because with our plants, because we already know that this plant is already genetic engineered to produce more products. Okay, so that can actually improve already the crop production. Now, another application of DNA is what we call the transgenic technique. Okay, so transgenic technique can be applied to improve the qualities of the crop. That includes protein content. Okay, so especially nowadays, some people who don't we have a limited resources in terms of proteins, and proteins are very essential in our biological activities, okay, inside of our body. So we can actually induce the protein that can be found in your plants if you don't want to consume other meat products. Okay, so we can also improve the amino acid composition, okay, of the food itself. Also, the starch composition. We don't need to rely to the rice. We can actually get this rice from other resources like the corn. Okay, and also the polysaccharide compounds. Polysaccharides, these are carbohydrates that we need in our body to generate energy. Okay, and of course, lipids, the fats that is essential in our activity that can actually boost our immune system. So these lipids can actually be found in seeds and other storage organs of the plants that help us, to improve the nutrition needed by a human. Okay. Now, we have the technology is actually improving, especially if we still are researching on what are the most effective ways, as well as eliminating those that need a large amount of time. Okay, so the development of modernization of agricultural based on new technology is an important choice, especially nowadays. And surprisingly, with modern DNA technology, it becomes more and more widely used in agriculture. Agriculture in terms of GMO, as we call it, genetic modified organisms, wherein we can address the shortage in terms of food. So the current major issues like food, population, and other problems that our world facing will get a better solution because of this new technology. And there are a lot of work to be done in some areas, such as techniques improvement, utilization of chromosomes engineering, especially in those innovation of crop germplasm, and also further increase of crop quality, and of course, biosafety prevention. These are the goals or tasks that is relevant wherein different researchers or group of researchers need to be focused on, especially nowadays. We already have the technology. All we have to do is to improve these technologies so that we can address our problem nowadays, which is the food shortage. And this is where I'm going to end my discussion. And by the way, these are the reference materials that I use to form all the data that I have here. Thank you so much for listening for today's lecture. Thank you.
Thank you, Miss, for your presentation. It's very interesting material for today. So, we can continue to discuss with our student. Jadi, Bapak Ibu sekalian, tadi sudah diberikan materi, tambahan materi dari Miss Christine, materi yang sangat menarik. Jadi, sekarang waktunya adalah bagi Bapak Ibu jika ingin bertanya kepada Miss Christine. Jadi, silakan siapa yang ingin bertanya pertama?
Saya, Bu.
Silakan Ibu Siti Rohmah.
Ya. Terima kasih. Thank you, Miss Kada. Let me introduce myself. My name is Siti Rohmah. I have a question about can genetically modified plants which have molecular implication for agriculture can have a negative impact such as making sick, killing leaf, lifting, and also destroying nature. Thank you.
Thank you for that. I didn't catch the first one. Chemical modified organism. My apology, but can you repeat again the question?
Let me repeat it, Miss. Miss Siti Rohmah is asking about can genetically modify plants which have molecular implication for agriculture can have a negative impact such as making sick to a human, killing living things, and also destroying nature.
Okay, thank you so much for that. For now, there is actually, in terms of negative impact to the environment, there are no researches, okay, published indicating the negative impact to the environment. The negative impact is in terms of ethical, okay, wherein the mindset of the people is that you can actually manipulate the characteristic of a product, okay. So in terms of the environment, at the moment, there's no recorded data, as far as I know, okay? And then in terms of human consumption, the genes, or the the manipulated genes, okay? They only targeted specific genes, okay? Indicating that there will be no impact to humans unless, okay, unless there is mismanipulation, if I call it, in terms of arranging the DNA structures, okay? Because there's a possibility that they will tap a certain gene that can actually sense by our body that will sense by our body that will change the biological activities if our body monitored that kind of protein. But again, before we can actually introduce this product to the market for human consumption, we already addressed this kind of negative impact. So we are aware that this GMO will not affect the human health, but more on addressing the challenges that we are facing nowadays. Thank you.
Oke, Miss, thank you for the answering. Jadi Bu Siti Roma, Miss Christine mengatakan bahwa untuk saat ini itu tuh belum ada berita yang bisa memberikan dampak pada manusia. Tetapi jika ada kesalahan saat memanipulasi karakternya, barulah timbul memberikan efek pada manusia. Tapi sebelum memberikan efek atau sebelum dipasarkan, mereka uji coba dulu ke tempat lain untuk mengetahui tingkat kenegatifan dari hasil produk transgenik tersebut. Oke, masih ada yang ingin bertanya? Is there any student want to ask to Miss Christine?
I am.
Baik, silakan Sylvie.
Okay. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
My name is Sylviane Papala. Thank you for the opportunity. I want to ask with Mrs. Dr. Mary Christine Chada from Trinity University of Asia, Filipina. Advances in plant biotechnology, particularly molecular biology, will provide opportunities to address and solve problems in plant breeding. How does molecular biology play a role in plant breeding? And how molecular biology techniques help understand disease in plants? Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you.
Waalaikumsalam warahmatullahi wabarakatuh.
I missed the molecular biology in terms of the second word. I forgot.
The question is two from Sylvie. The first one is, how does molecular biology play a role in plant breeding? And how molecular biology technique help understand disease in plants?
For the in terms of plant diseases, okay, so this is actually based on my previous research. First, we isolate the pathogens, so especially fungal pathogens like Fusarium. So we identify the DNA structure of that Fusarium species, and then we identify the specific gene, okay, responsible for that infection. From there on, when we identify that specific gene, we try to eliminate, okay, that gene responsible for infection, so that the Fusarium, not as pathogen, will be used in other form, like anti-cancer. We also have anti-tuberculosis as we perform it, and also in terms of using as biofertilizer. Okay, we test, we screen, okay, that kind of Fusarium, that pathogenic Fusarium, okay, and then we test another Fusarium wherein we eliminate the gene responsible for the infection. Okay, so wherein if we perform antagonistic activity. If this Fusarium that doesn't have the gene for infection can actually combat the Fusarium that has pathogen that has the gene for infection of plants. Okay, so I hope I answered the first question. My apology that I missed the second word. What is the molecular impact? Molecular technology impact? The second word, I forgot. The first question, sorry.
Okay, the first one question is, how does molecular biology play a role in plant breeding?
Okay, thank you so much for that. Okay, so here in plant breeding, okay, so first we want to identify a specific trait that we want to work on. Okay, so for example, we want to produce tomato that will not be infected like Fusarium. Fusarium is the common pathogens for that plant. So what we did is that for the tomato, we identify the DNA structure and identify the specific gene that usually targets by the Fusarium pathogen. Okay, so when we eliminated that gene, okay, we can actually produce another seed, that doesn't have that gene responsible for infection. So if we already eliminate that one, we can actually mass produce those tomato seeds that will increase the tomato seeds that is good for plant growth. Okay, so at the same time, tomato, we want, of course, in terms of the appearance, we want them as have a good color, no, as well as the shiny color. Okay, so we actually we are now into that kind of process wherein we are improving the appearance of the tomato so that the what you call this, the farmers here in the Philippines can produce this kind of products and can be export, no, in other countries as well. But we are on the early stage on that matter.
Okay, thank you for the explanation. Jadi Sylvie, Miss Christine tuh mengatakan untuk pertanyaan pertama sama pertanyaan kedua itu intinya sama. Jadi misalnya kita memilih tomat, tomat itu kita ingin memilih misalnya ada apa penyakit di tomat tersebut. Kenapa penyakit itu menyerang tomat? Nah, dari situ kita mengambil gen yang bertanggung jawab mengapa tomat itu buruk. Jadi kita ingin hilangkan. Kita sudah mengecek gen-gennya yang mana, terus kita mengambil misalnya gen BT. Dari itu kita bisa tambahkan gen BT ke tomat untuk menghilangkan penyakit yang ada pada tomat. Dan setelah kita gabungkan, setelah kita combine, terus dipilih markernya dan kita tes di lapangan. Jadi dari situ, kalau misalnya kita merasa cukup, itu barulah kita bisa produk itu bisa dipasarkan ke masyarakat luas. Itu untuk teknik dan bagaimana biologi molekuler itu berperan dalam breedingnya, dalam breeding tanamannya itu. Bagaimana, Sylvie?
Thank you, ma'am, for explanation.
Okay, is there any student want to ask?
May I ask to Miss Christine Kada about the application of biotechnology in agriculture can assist in accelerating seed production, improving plant properties so that can produce superior new types of plants. Do genetically modified plants behind their advantage have negative traits for humans and animals that consume them? And according to you, how is the development of transgenic crops in the world today? Thank you.
Okay, so in terms of the transgenic crops, transgenic technology now in different parts of the world. So in Asia, we have in Korea as well as in Japan, wherein they are now into that level of using the technology, to improve their products. Let me rephrase that answer. In Asia, Central Asia, as well as in some countries in Europe, they are now into that one, okay? Because first they need to address the lifespan of fresh produce. When we say fresh produce, these are crops, vegetables, and fruits, okay? So first, they want to improve the length or the lifespan of the vegetables, okay? That is actually they are already available in their market. I experienced that one in 2009 when I was in New Zealand. You can actually, they already use the technology, okay, wherein you don't need to, you can actually store the fresh produce for more than a week inside of your refrigerator, or if you have those vegetable storage inside of your home. You don't need to buy every now and then fresh produce from the markets. It actually gives you an ample time to do other things. Okay. So at the same time, when I visited the farm in New Zealand, as mentioned, they don't rely already on chemical products like fertilizers, pesticides, so on and so forth. And it also indicates, they already addressing the challenge in terms of food production. They don't rely in importing fresh produce from other countries because they already improve their food products, okay, using this transgenic technology. Okay. However, in our country, like in my country, in home country, we are still in earlier stage on using this product. Although we already apply this in terms of rice, because rice is a staple food in our country. So we're in the rice, if you're going to cook it, it will take time before it becomes spoiled. Okay, before it actually exposed to bacteria. Okay, so it will actually, you still have a food inside your house. No? You don't need to go out, take time to do your grocery every now and then. So yeah.
Oke, thank you, Miss. Jadi Canda, pernyataan dari Miss Christine tadi itu sama dengan hampir sama dengan pertanyaan dari Bu Siti Roma tentang konsumsi untuk manusia dan hewan. Jadi memang kemungkinan itu nanti ada setahap negatifnya, tapi pasti akan diuji coba dulu sebelum dimulai dipasarkan. Nah, kalau untuk bagaimana perkembangannya saat ini, itu mulai dikembangkan terutama di New Zealand tadi. Jadi sudah beberapa masyarakat di sana itu sudah dapat sudah mulai menikmati produk ini tanpa harus lagi mencari-mencari di supermarket yang ada. Jadi mereka sudah bisa dapat di mana-mana, terutama di pasar, mana dan di mana saja. Oke, apakah masih ada pertanyaan lain untuk Miss Christine? Is there any question?
Okay. Miss, I excuse to ask a question. Before that, maybe I can introduce myself first. My name is Rusma Abrianti, and nice to see you in this and our discuss. Thank you very much for your matter. It's very interesting. Maybe my question is almost same with another friends. That I want to know about when we or us consuming a product of GMO, maybe it get a negative impact. So maybe we can also, what can be done to avoid the impact of consuming genetically modified plants. It's only how we can solving the problems when we consuming genetical products or plants.
In terms of the problem, in terms of what? I guess I missed something on that part.
This only sometimes I have to heard when we consuming example soybeans, we can get allergen, especially for traditional food in Indonesia like tempe. Now, so maybe you can give me or my friend or anything in here knows about the new knowledge, maybe how we can solving the problem, or maybe we can get a new solution how to the allergen.
Okay. In terms of for example, as mentioned, we have that here in the Philippines in terms of mango. Okay, so there are people here in the Philippines that they cannot actually eat mangoes, okay, because they also secrete a substance that is considered as allergens to those immunocompromised individuals. Okay, so first, it's actually a two-way street. So first, the researchers need to identify the specific cells or marker that is responsible for the allergens against the mango. Okay. Now, next, you need to identify from the mango using the DNA the specific gene that will trigger as an allergen to those immunocompromised patients. Of course, our body, we cannot actually manipulate it. So what we can do is from the mango. Okay, either you have two ways from that mango. You eliminate, you're going to identify the genes and eliminate it and produce another seed that doesn't have that allergens. Okay, so if you cannot do that, what you're going to do is to find another genes from other cellular organism, maybe a fungi, bacteria, or other plant resources, wherein the gene can actually combat the allergen protein or gene marker inside of that mango. Okay, so why I am keep on mentioning bacteria, the fungi in terms of this kind of product, okay? Because the microorganisms, they have different applications. And then at the same time, they are environmental friendly. Environmental friendly because you don't, if you're going to mass produce it, all you have to do is to place them in their substrate. Okay? Substrate indicating their food. You need to just mass produce it using that liquid. That's it. But if you're going to for example, use another genes from another plant, you need to kill that plant. And that will a problem in terms of our environment. We need to preserve what we have in terms of our plants. Okay, so that's the reason why we have, I don't know if you already aware of this, because I find numerous researches in terms of endophytes. Okay, so endophytes, these are microorganisms that actually live within the plant tissues. They have a symbiotic relationship. Okay, they have a give and take relationship with the plants. So you don't need to kill a certain plant organ to study them. All you have to do is to isolate the microorganisms that live within that plant tissue, and you can work anything on it. So that is the two options, no, in terms of your problems in terms of allergens.
Okay, very interesting explanation to avoid this allergen. Okay, Rusma, gimana?
Yeah. Thank you very much for the answer, Miss Canada.
Thank you, Ashley.
Oke, silakan untuk satu pertanyaan lagi.
Izin, Bu.
Silakan, Reno.
Okay. Thank you, Miss Shada, for opportunity. Okay, in the case of modif fruit with no seeds, to make it easier to eat, how is the process of biodiversity continuation of the species from these plants, and how is the impact of the farmers in the nursery process?
Pardon, impact of the
Farmers in the nursery process.
Okay, in terms of biodiversity, I don't know if you are aware of this one. Before we can actually do genetic modification with the plants together with the seeds, we need to store them in the seed bank. That can be found in somewhere in Europe. I think that was in Poland. Okay, so we need to deposit, okay, the original seed as well as germplasm, okay, before we can actually modify it. Okay, so in terms of biodiversity, we can actually preserve it. Okay, so we do like freezing. I forgot the term. Not by freezing, no, but we actually, what you call this? We have those ampules wherein we're going to place them in a certain liquid and then put the seed itself and then let it store for about for a long period of time. Just make sure the temperature is about -10 degrees down. Okay, so we can preserve that one in terms of biodiversity. Okay, so at the same time, if we want to keep on using these products, okay, we are actually allotted a certain greenhouse, okay, greenhouse in a sense that we can actually plant them also in the forest or other places in the Philippines as well. Okay, so because in our group we do field not field trip, but field observation to identify if there are plant species that can be found in a certain forest or mountains or a certain plateau. Okay, so that is my answer for the biodiversity. Another one is the impact in terms of the farmers. Am I right? Nursery process. Okay, we actually let them first inform what we're going to do with the certain product. So for example, last month, I think that was last month, the farmers in Laguna, it is a place where we usually have those plant cultures. Okay, so we inform them what we're going to do in a certain plantation wherein we rent it to observe a mass production of a certain anti-vaccine for the plants. Okay, so plants also have viruses. Okay, so we inform them every week, okay, what we're going to do next and what are actually their comments, suggestions in this kind of activity. Because if we're going to listen to our farmers, we can actually learn from them. We have information that we don't know, but yet they don't know if we're going to compare between these two people. So, and then at the same time, you need to, if you're going to propose something to them in terms of research, you need to give them a product, a sample, so that they will be oriented what will be the output of your research or activity. So it's a two-way street. Okay, it's you need to meet halfway on how you're going to address those challenges. You?
Oke, Reno. Jadi kata Miss Christine itu mengatakan bahwa kalau untuk di petani, jadi yang penting itu informasi. Jadi informasi itu sangat penting untuk keberlanjutan dalam proses nursery prosesnya. Jadi informasi dari waktu ke waktu dari si peneliti ke farmer itu sangat penting, informasi itu terus berjalan. Dan itu akan sangat sangat penting untuk biodiversitasnya dari tanaman yang tidak memiliki biji untuk dapat aman dimakan oleh manusia. Oke? Gimana?
Okay, thank you, Miss Sarah.
Okay, Miss, thank you for your presentation and for your answering the question from our student. It's very interesting to hear about implication biology molecular in agriculture in Philippines and other world and another country in this world. So now it's Miss Christine's turn. Does Miss Christine have an assignment for our student?
Actually, I did not prepare anything for that matter. So, yeah.
Okay, it's okay. It's no problem. But can you share your presentation, your Microsoft PowerPoint to my in the chat room or via Miss Eri Agusta? It's okay. Then the list course attending list is already I sent to you in the inbox chat. You can click and you can write it this course attending list.
Oh, sure. Yes, ma'am. I can share the PDF here in our Zoom, right? I can actually...
Okay. You can share.
Yes.
You can share, Miss. Thank you.
I'm just saving it as PDF, so I can actually place that here in our Zoom chat. Let me just check if it's already available.
Okay, it's okay, ma'am. No problem. So, sambil kita menunggu Miss Christine untuk memberikan materinya, jadi berikutlah hari ini itu, jadi mata kuliah kita dari Miss Christine mengenai implikasi biologi molekuler untuk di bidang pertanian. Nah, Miss Christine ini akan memberikan materinya tadi lewat chat room, nanti Prodi yang akan memberikan ke teman-teman sekalian dan juga lewat saya, jadi bisa kirim ke grup. Nah, kebetulan untuk tugas dari Miss Christine itu belum ada. Jadi apakah nanti akan ada, kurang tahu juga. Tapi jika memang ada, nanti saya akan memberitahukan kembali kepada teman-teman sekalian. Okay, thank you, Miss, for the file. Let me save it first.
We have a certificate for Miss Kada.
Oh yes, to certificate for attending this course from our student from our study program to Miss Christine. We have a certificate for you. Graduate program has started screenshot. This is certificate for you from us.
Thank you so much for that.
Thank you.
We are very thank you for your time and for your
thank you
yes.
for the opportunity.
Jadi berikutlah selesai mata kuliah kita hari ini dari Miss Christine dan sangat berterima kasih kepada Miss Christine untuk pemberian kuliahnya hari ini. Jadi before we close today lecture, I thank you to Miss Christine for delivering very interesting material, and I hope very hope that Miss Christine can become a guest lecturer again in the future in other course in our study program. Thank you, Miss Christine, for this opportunity, and thank you for our students for attending these courses for today. Okay, study program, do you want to take photo for this today lecture for today?
Okay, 1, 2, 3. Okay, thank you.
Thank you, Miss Christine, for your attending for today, and thank you for the time, and thank you for everything for today. This is very first time me as moderator in this courses in other country. Jadi I'm very nervous to miss from the today courses. Jadi terima kasih teman-teman semua atas kehadirannya hari ini. Jadi kita sangat beruntung mendapatkan materi dari Miss Christine hari ini. Jadi kita mendapatkan tambahan ilmu, tidak hanya ilmu dari Indonesia, tetapi kita juga mendapatkan ilmu dari dosen di Filipina. Okay, thank you for your all today time. So I will close today this lecture. Assalamualaikum warahmatullahi wabarakatuh, and good afternoon for you all.
Good afternoon.
Thank you, everyone.
Nice to meet you.
Thank you, Miss.
Thank you, Miss.
Thank you,
Miss.
I will leave now. Thank you so much for the opportunity. Thank you.
Okay, thank you.
You're welcome, Miss.
Maaf.
Sekianlah kuliah kita hari ini. Jadi sangat beruntung kita mendapatkan materi dari Miss Christine. Jadi untuk saat ini, karena mata kuliah kita juga sudah selesai, apakah kalian sudah download semua materi dari Miss Christine?
Sudah, Bu.
Sudah? Oke, nanti saya menanyakan ke Pasca, apakah nanti tetap akan ada tugas dari Miss Christine atau tidak. Oke? Tapi jelas hari ini terima kasih atas diskusinya tadi, walaupun saya sudah membuat urutan untuk siapa yang bertanya, tapi saya sangat menghargai bagi teman-teman yang berani bertanya tanpa harus disebut namanya satu-satu. Salut kepada Bapak Ibu sekalian. Jadi karena waktu kita juga sudah selesai, terima kasih atas waktunya. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Terima kasih.
Saya izin pamit ya. Sama-sama, saya izin pamit.
Oke, untuk ekofisiologi tumbuhan dan Oke, untuk ekofisiologi tumbuhan dan hewan, mungkin saya konfirmasi dulu ke Bu Yeti, apakah sudah bisa dimulai atau nunggu jamnya? Untuk yang belum maju, siapa saja yang belum?
Aku, Kak.
Oh iya,
Banyak sih, Kak.
Canda. Pertemuan terakhir ini.
Hmm.
Pak Julian sudah belum, Pak?
Udah, Pak. Pertama kali kemarin.
Oh iya,
Pembahasannya
lupa.
beda.
Iya.
Ya, makasih,
Yang
Pak.
belum tuh, Kak. Aku, Mbak Silvi, Mbak Reli, Mbak Yeni, Mbak Ica, Mbak Jurgia.
Oke. Lumayan banyak ya.
Baru 6 yang belum. Bu
Yang
Siti, belum kan?
Oh iya, enggak jadi, Bu. Kemarin.
Allah.
Yang sudah ada di Zoom ada 3, Bu Siti Rama, Canda, dan Silvi. Oke, dikonfirmasi dulu. Tidak apa-apa kalau nak off sambil ke yang lain. Kita mulai.
Baik, Pak.